And the reason I wanted to stop for just a moment is we're going to start thinking about a different group, about the fungi. So I want to stop here for a minute and so you should see the page titled fungi now. If you don't see that, let me know. And it means that I am showing you the wrong thing. So right now there are about 150,000 species of fungus that have been identified and classified and studied. Most scientists think there's probably about 20 times that, somewhere between 2 and 3 million species. Why haven't most of the fungi been studied? Because as far as we know, they don't have any industrial use. They don't have any impact on human health. They don't have any impact on the health of plants or animals that we use in agriculture. So they don't seem to have much interaction with people. However, one of the things that we definitely know from our past studies, not just of fungi, but of other groups, there are probably a lot of these fungi that haven't been studied yet that do impact human existence. It's just that we don't know how yet. So we want to keep that in mind. So let's look at some of the general characteristics of fungi. Well, one characteristic of fungi is fungi all have eukaryotic cells. They're in the kingdom Eukarya. Most fungi are unicellular. There's one big group called the yeasts that are single-celled. Fungi have cell walls like bacteria did, and like we haven't gotten to them yet, but you probably remember from 105, like plants do. Now notice bacteria had cell walls made of peptidoglycan. And plants, as we'll see, are going to have cell walls made of cellulose. Fungi have cell walls made of chitin, so a completely different chemical. So again, if I gave you a chunk of cell wall, you could analyze the chemistry of that piece of cell wall and you could figure out whether that came from a bacteria cell or a fungus cell or a plant cell. Most fungi have filamentous bodies, and filamentous just means thread-like. So if we look at the filamentous body that we see, what we see is that there are long strands of cells. Sometimes these strands are packed very tightly together. Sometimes there are spaces between these strands. Now, when we're looking at these cells, notice that these cells have a little purple dot in them if we look at this strand on the left, and this represents the nucleus. In most fungi, if you're looking at a mold growing on a piece of bread, or if you're looking at most other type of fungi that you would see, if you're looking at some athlete's foot on your foot, the cells that you're looking at are probably haploid cells. The haploid stage of the life cycle is the dominant stage. Notice dominant just means this is the stage of the life cycle that they're in for the longest period. This is what we usually see when we're observing them. So kind of like when we talked about dominant in genetics, doesn't have the word that we necessarily associate with dominant out of the scientific circles. So at some point during the cell, haploid cells fuse and we get multinucleate cells. First, we get cells that have more than one haploid nucleus. So the other thing that we see that's a characteristic is we have a cell wall, and the cell wall that extends between one cell and another cell when we're looking at a filament. This is called a cross wall or a septum. And when we talk about fungi, they fall into two general categories. If we talk about septate fungi, they have cross walls that separate one cell from another cell. If we talk about non-septate, or it's probably more common for these to be called aseptate cells, there are no cross walls. So you see lots of nuclei in a single cell. The cells are multinucleate because this cell was really formed by numerous cells fusing together. Now, a single strand, or filament, whether it's septate or aseptate, non-septate if you prefer, is called a hypha. And if we look at all the hypha that make up the fungus's body, all the hyphae together are called the mycelium. So we're going to see mycelium and hyphae a lot. So, when you see hyphae, think of a single thread. A mycelium is dozens or hundreds or thousands of these threads and how they're twisted around each other. When we look at the fungi don't have flagella, except one group of them. And the group that does have flagella was actually classified with Protista until very recently. And the reason they were classified with Protista, one of the main reasons, was because they had flagella. And fungi were defined as not having flagella. But going back to what I said before, guess what DNA analysis shows, as well as metabolic analysis of some of the processes in the cells? It shows that they're really more closely related to fungi than they are to Protista. So they used to be a group of Protista. Now they're a group of fungi. Now, when we look at fungi are described as being heterotrophic by absorption, and this is what's called their mode of nutrition. When we look at organisms, their mode of nutrition, they're either autotrophic, usually photosynthetic, not always. They're heterotrophic by absorption, like fungi, or they're heterotrophic by ingestion, like animals, like us. Well, if you're heterotrophic by ingestion, you eat your food. You take a bite out of it. If you're heterotrophic by absorption, we were just talking about digestive enzymes a moment ago. After you eat your food, you start releasing digestive enzymes on it. There are some weak enzymes in your saliva. There are more enzymes in the stomach. There are more in the small intestine. If you ever want to test the weak enzymes in your saliva, there's a real simple science experiment you can do at home. If you get something like a saltine cracker that's almost completely carbohydrate, take a bite out of that saltine cracker, chew it up, takes a little bit of patience, chew it up for a long period of time. Let it kind of sit in your mouth and get nice and soggy. And if you can avoid just being grossed out, one of the things that you can observe after a while is the cracker starts to taste sweeter. And this is because most of the carbohydrates in a saltine cracker are polysaccharides. But the enzyme that's found in your saliva, salivary amylase, takes starch, what the carbohydrate that's found in the cracker, and breaks it down to disaccharides. And disaccharides taste sweet. Starch doesn't taste sweet. So if you chew up a cracker, just let it sit in your mouth for maybe 5 minutes. Kind of gross, it gets soggy, but it'll taste sweeter. Now, if we look at fungi, a lot of fungi are saprophytic, meaning they decompose dead organic material. So they take those complex organic molecules, like we were talking about a moment ago, and break them down into simpler molecules, which the producers in an ecosystem, things like plants or photosynthetic algae or photosynthetic bacteria, those simpler nutrients can be used. There are some fungi that are parasitic. We talk about yeast infections, we talk about jock itch, we talk about ringworm. All of these are diseases caused by fungi. Some people have fingernail fungi or toenail fungi, I should say, that make their toenails kind of thick and yellowish. Now, notice that fungal diseases are going to be much more difficult to get rid of than protistan diseases or bacterial diseases. Why? Well, go back to that cladogram that we had a minute ago in screen sharing. Fungi are pretty closely related to animals. So this means it's pretty hard to find antibiotics that affect fungal cells that don't affect animal cells, like your cells, for instance. So fungal infections can be very difficult to get rid of. Notice that fungi are the only eukaryotic organisms that make enzymes that can break down a chemical called lignin. And in 105, we mostly talked about cellulose in the plant cell wall, but there are several chemicals. One of them is called lignin. And so fungi can break this down. And there are actually a few species of fungi that are carnivorous because of this kind of net that we see when we have lots of hyphae to make up that mycelium. There are some fungi that actually live in the soil and they trap small worms inside their mycelium and then they digest them. Now notice when we say that they're heterotrophic by absorption, a lot of the enzymes that fungi produce to digest their food are the same enzymes that we find inside us. But if we look at animals, most animals digest their food either inside the cells of the body or sometimes inside a chamber in the body, like the stomach, for instance, or the small intestine. If we look at fungi, fungi release their enzymes onto the environment that we're living on and they liquefy it. This is why if you've ever picked up a piece of bread that has mold on it or a piece of fruit that has a mold on it, if the mold's been on very long, if you pick up the fruit, it's all squishy or the bread is squishy because it's basically breaking down the proteins in the bread into amino acids. It's breaking down polysaccharides into monosaccharides. It's breaking down triglycerides into monoglycerides and fatty acids. So basically they're taking big molecules, and again, these are hydrolysis reactions, breaking them down to small molecules. So they do the same type of digestive reactions that happen in our digestive system. They do them outside of their body though, and then they absorb these liquefied nutrients. So when we say heterotrophic by absorption, the digestive enzymes are released into the environment, break down the food, and then the nutrient molecules are absorbed into the cells. So there are 5 major groups of fungi, although one of the things we see is that, all the people who study fungi don't agree on these 5 groups. The further we get away from humans, from mammals, the more controversy there is, because when we look at mammals, mammals are pretty similar to people. Biologists can look at them and kind of agree at least on the main categories. But when we look at things like fungi, they're so different from human biology. Remember what I keep saying, the domain they're in has a biological meaning. They're eukarya because they have eukaryotic cells. But the other levels of classification are arbitrary. So there are 5 main phyla: the chytridiomycetes, the zygomycetes, the glomerulomycetes, the ascomycetes, and the basidiomycetes. And notice that they're sometimes called the zygomycota, the ascomycota. Mycetes and mycota both mean fungus. That's why a lot of things that resemble fungi are going to have that prefix. So as we talk about different parts of the fungus body, a lot of them are going to have MYC somewhere in the name. Now, how do we put them into these different categories? Really, besides their DNA, now we're talking about organisms that are large enough. We can look at them and we can use their morphology. They can use their anatomy. So we look at the type of sexual reproduction they have. And we also look at the degree of separation of the mycelium into the cross walls. So, you know, we're going to talk about septate fungi that have those cross walls, aseptate fungi that don't. So almost all the fungi that we'll look at are multicellular fungi, and their body is called a mycelium. Notice there's that myce part again that refers to fungus. And it's made of filamentous algae. Now, the algae are necessary because notice if you're going to absorb your nutrients from the outside environment, it takes a large surface area. So let's look at why surface area is important. And we're going to go back to a little bit of math. This is math that you probably looked at many years ago, but you may not recall. So let's think about a cell that's a perfect cube. Now, I recognize that cells aren't perfect cubes, but keep in mind the math gets much more complicated if we look at a cell that's oval. And let's look at a cell where each side of our cube is A units. So if we unfold our cube, so here's, you know, the side that was on the left, the side that was on the right, the top, the front, the back, the bottom. If we look at our cube, what we see is that the area of each side, right side, left side, front side, back side, top side, bottom side, the area of each side is A squared because it's A across, it's A down. So we know that the area of a square is length times width. Now, since we have 6 surfaces on a cube, that means our total area is 6A squared. Now, when we're talking about something like this cube, or if we're talking about an animal, this is called the surface area. And surface area is important conceptually because when we think about surface area, this is proportional to the area of the plasma membrane. So if you're thinking about a cell, well, the plasma membrane is really the only place where a cell can exchange materials with its environment, either bring things into the cell or eliminate waste products that it doesn't want. So when we think about surface area, this is basically the plasma membrane size. The other thing that we have to think about is the volume. Well, volume is length times width times height, or A times A times A. So in this case, that's A cubed. Now, let's think about putting some numbers here instead of just arbitrary units. So let's do this like an algebra problem. Let's let A equals 2. So if A equals 2, then our surface area is 6 times A squared, 6 times 2 squared, 24 square units. We haven't specified the units, so we could be talking about millimeters, centimeters. Our volume is going to be A cubed or 2 cubed, which is 8. Now, what we're interested in is not the surface area by itself or the volume by itself. What we're interested in is the ratio of surface area to volume. So in this case, the surface area was 24. The volume was 8. So if we take 24 divided by 8, a ratio is a fraction, we get 3. Now think about what this means from a practical standpoint. This means there are 3 square units of our plasma membrane for every 1 cubic unit of cytoplasm and organelles. And we can think of the surface area as being equivalent to the plasma membrane. The volume, since this is where we have cytoplasm, organelles, metabolic reactions occurring, the volume is proportional to metabolism. So basically this means we have 3 units of exchange area for every 1 unit of metabolism. So let's see what happens if we take our cell, and let's stick with a numerical example. Let's double the size of our cell. So up above we said, let's try this with A equal to 2 units. So let's try this with A equal to 4 units. And let's look at 2 different ways of thinking about this. Let's take our cell that was 2 by 2 by 2, and let's just double all the dimensions of our cell. So if we take our cell that's 2 by 2 by 2, the surface area of this cell is going to be 4 by 4 times the 6 faces, or 96 square centimeters. They're using centimeters here. Pretty big cell if it's 2 centimeters across. Now, let's think about the volume. The volume of this is length times width times height. So 4 times 4 times 4. Well, 4 cubed is 64. So our surface area to volume ratio is 96, our surface area, divided by 64, our volume. It's 1.5. Well, to remind ourselves, let's go back up here and notice that our surface area to volume ratio, when the dimensions of our cell were 2, our ratio was 3. Now, if we double it, our ratio is 1.5. So what does this mean? Well, it means that we don't have as much plasma membrane for every unit of cytoplasm in here. So our cell is twice as big. We don't have as much plasma membrane per unit of volume to either bring stuff into the cell, reactants that we might need, or to eliminate waste products. So what's the solution to this? Well, the solution is, let's chop this large cell into 8 small cells. Let's cut it down the middle in the front. Let's cut it down the middle from right to left. And let's cut it down the middle from top to bottom. And we'll end up with 8 cells that are 2 by 2 by 2. Basically, we're going to end up with 8 copies of the cell that we had here, 2 by 2 by 2. Okay, now our surface area for each cell, for each face of each cell, I should say, is 2 by 2. There are 6 faces, so that's 24. Now, notice if we go back, that's the same thing that we had back here, which makes sense because our cell is the same size, but we've got 8 of these cells. So our total surface area is 8 times 24, 192 cubic centimeters. What's the volume? Well, the volume is still the same. The volume is 2 by 2 by 2, 8. And then we have 8 of those, so 64. So under this new circumstance, what do we have? If we have small cells, we have 192 square centimeters for the surface area. If we have one big cell, we only have 96. So you can do the math, but basically if you do the math, since we have 8 cells that are all the same size as we had before, you get the same 3-to-1 surface area to volume ratio. So what's the whole point of going through this mathematical exercise? This is why as living organisms get bigger, they don't just want to have larger cells. If you have bigger cells, the bigger the cells are, the less surface area, the less plasma membrane you have for each unit of volume, for each unit of metabolism. So what does that mean? That means it gets harder for materials to enter the cell and leave the cell. So if you think about this in terms of 2 gases that living organisms need, oxygen and carbon dioxide. If we're looking at a plant cells are constantly using up carbon dioxide in the Calvin cycle, or the dark reactions of photosynthesis. They take carbon dioxide and it's gradually converted to glucose. So this means the concentration of carbon dioxide in a plant cell is low. So carbon dioxide will diffuse into plant cells. On the other hand, one of the products of photosynthesis, remember that non-cyclic photophosphorylation, one of the products of that was oxygen. So plant cells are constantly making oxygen. So oxygen is at a high concentration inside plant cells. It's at a low concentration outside. So oxygen is constantly diffusing out. Plants use a little bit of the oxygen they make for aerobic cell respiration, but they make so much that most of it is just a waste product that they eliminate. So if plant cells get too big, what happens? It's harder for carbon dioxide to get in, and it's also more difficult for oxygen to get out. So as living organisms get bigger, it's better to be multicellular than to just stay single-celled and have one immense cell, because that one immense cell may have problems exchanging materials with the environment. Now, this is such a basic principle. We can see this surface area to volume ratio everywhere we look when we study biology. If we look at elephants, elephants are like people, they're warm-blooded, except elephants have a problem. Elephants' body is so large that they tend to retain too much heat. And since in nature, elephants live either in Africa or in India, areas that are relatively hot, they've got to have a way to eliminate heat. So how do they eliminate heat? You ever seen the ears on an elephant? The ears are huge and they're very thin. And what you might not know is they have a very rich blood supply, because blood transports things around the body. One of the things that blood transports around the body is heat. So by having a lot of blood vessels in their very thin ears, this means that these blood vessels are in very close contact with the environment. So this means that heat will move from the elephant's ears, where it's at a higher concentration, the temperature is higher, in other words, to where it's at a lower concentration. So this is a way of dissipating heat. Some people think when you look at dinosaurs like Stegosaurus that had those plates on the back, that may have been one of the functions of those plates on a Stegosaurus. Notice that we see the same thing to varying degrees when we look at other characteristics. If you think about people, if we make measurements of different parts of the body in people, one of the things that we see when we measure Native Americans, like the Inuits, sometimes they used to be called the Eskimos. But when we measure them, if you compare people who live in Greenland to people who live in Africa, people in Greenland tend to be shorter and thicker. Well, why would it be advantageous if you live in Greenland to be shorter and thicker than if you live in Africa? Well, because if you live in Africa, the climate is warmer. If you're more slender, that means you have more surface area per unit volume. That means it's easier to eliminate excess heat. If you live in Greenland, you don't want to eliminate excess heat. You want to hold on to that excess heat. So there have been studies showing that people who marry within their tribes in those ethnic groups are still relatively shorter and stockier than other people's. Of course, there's a lot of intermarriage with different groups now, so that's not as pronounced as it used to be. If you look inside the human body, you can see the same thing inside the human body. If you look inside the human body, if you look in our lungs, for instance, our lungs don't have a nice smooth inner surface where oxygen diffuses out of the air we breathe and into our blood. If we look inside our lungs contain a lot of tiny microscopic structures called alveoli. The alveoli kind of look like little grapes. Why have all these little alveoli instead of a nice, smooth, hollow lung? Because all those little alveoli give a lot more surface area. So that means we can get more oxygen into our body quickly. We can also eliminate more carbon dioxide from our blood quickly. And notice, if we think about surface area and volume, the reverse also applies. If you look at plants like cactus, cactus plants live in hot, dry environments, and plants lose most of their water through their leaves. So the leaves on a cactus are the needles. Why such small, skinny leaves? Because there's not much surface area. So if there's not a lot of surface area, that means that they don't lose as much water through their leaves as a plant like an oak tree or a maple tree would, with much broader, thicker leaves. So we can even think about the reverse. Sometimes when we see very narrow, very thin structures, sometimes that's to minimize exchange with the environment. Now, since fungi are heterotrophic by absorption, this means that having a lot of those thin, skinny hyphae, and they're often separated from other hyphae, so they're not right next to them. A lot of times they're packed pretty loosely. Having them packed loosely, having a lot of those thin hyphae, that's an advantage if you have to absorb your nutrients instead of having a big blobby body, because you have more surface area for unit volume. Now let's look at one more aspect about fungi before we leave for the day. And that is, let's think about the fungus life cycle. So let's magnify this just a little bit more. And where we want to start is right in here in the middle with the mycelium. And the mycelium is the fungus body. Sometimes it's packed pretty tightly with hyphae, like if you look at a mushroom. Sometimes the hyphae are packed more loosely, like in a mold. Notice that the cells in a mycelium are N, or remember another name for N, and we could say 1N, but another name for this is they're haploid. Well, the mycelium can produce structures that make spores, and it makes these structures by mitosis. Notice this says spore producing structure because depending on the group of fungus, the spore producing structure and the actual spore often have different names. But since this is made by mitosis, all the cells in here are still N. They're still haploid. This structure makes spores by mitosis. The spores are haploid or 1N. And these spores, again, drift on the wind. Sometimes they float in the water. And when they land in a suitable environment, they germinate and start growing. And when they grow, they divide by mitosis and they make a mycelium. Now notice that, similar to what we talked about earlier with our slime molds, for most fungi, when conditions are good, they reproduce in this manner. And notice that this is a form of asexual reproduction. Asexual reproduction means the DNA isn't changed when we make a new individual. So unless there are some sort of mutations when we make these spores, and there could be because we do have to replicate DNA to make the spores, but unless there are some types of mutations, all these spores here are genetically identical to the mycelium they came from. And when they start a new mycelium, that mycelium will be genetically identical. However, every once in a while, fungi will reproduce sexually. So let's see what's involved with this. Well, there are different types of hyphae in a fungus species, and the different types are called mating types. They used to be referred to as males and females, and then at some point biologists realized that, you know, we were kind of trying to impose human norms on fungi. There weren't really males and females in any sense of the word. So now they're just called mating strains or mating types. So we have hyphae from two compatible mating types that make contact. And when they make contact, the first thing that happens is called plasmogamy. So think of the beginning of that word. It comes from cytoplasm. Plasmogamy means the cytoplasm of the two strains comes together. Now, when the cytoplasm of the two strains comes together, notice at first, the nuclei stay independent. So what we have is a mycelium, but this mycelium is now referred to as heterokaryotic. Hetero means different. There are two nuclei in it. And we describe its genetics as N plus N. We don't say 2N while it's heterokaryotic. And notice heterokaryotic is sometimes also referred to as dikaryotic because di means two. Now, after a period of time, the two nuclei fuse. Notice these nuclei are haploid. So when they fuse, they're going to make a zygote, and the zygote is 2N or diploid. And the fusion of the nuclei is karyogamy. So notice the cytoplasm fuses first, then the nuclei fuse. And once we have the zygote divides by meiosis to make the sexual spore reproducing structure. Well, we have a diploid cell dividing by meiosis, so we're going to get haploid cells, cells that are 1N again. And this makes spores that are N. These spores germinate and divide by mitosis and make a mycelium. However, notice when we follow this path, we don't necessarily get a mycelium back here at number 7 that's genetically identical. Why? Because after the nuclei fused, we made spores by meiosis. So think about looking at an individual organism that could reproduce this way. So suppose you could do this. Well, imagine that for a characteristic where you're heterozygous, some of your spores would get the dominant allele, some of your spores would get the recessive allele. So all of these spores are not genetically the same. So in most organisms, when we look at their life cycle and we're trying to characterize it in terms of the chromosomes, we have two possibilities. Part of the life cycle is haploid, 1N. Part of the life cycle is diploid, 2N. But with fungi, we also have a part of the life cycle that's heterokaryotic, that we have different nuclei in the same cell, so within the same cytoplasm. Now notice, even when we look at sexual reproduction, the actual part of the life cycle where we have heterokaryotic cells or we have diploid cells is actually a very short period of time. So remember what we said at the beginning of our discussion of fungi a little while ago. When you look at a fungus, the cells that you see are mostly haploid cells. One of the big misconceptions, and unfortunately Biology 105 kind of feeds into that, you know, when we do genetics at the end of Biology 105, what do we talk about? We talk about a person making gametes or a cow making gametes. And we talk about organisms that are diploid, they make gametes that are haploid. The egg and sperm come together and we have another diploid cell. If we look at fungi, almost any fungus that you see is going to be haploid. If we look at bacteria, bacteria are all haploid. They're never diploid. They have one copy of each gene. So with bacteria, we can't even talk about genetically something being dominant or recessive, because since they only have one copy of a gene, it always shows up in their phenotype. When we look at Protista, most of the Protista have a haploid stage in their life cycle. A lot of them have a similar life cycle to fungi where they're really only diploid for a short period of time. So one of the things that we want to keep in mind when we think about living organisms, the idea that most living organisms are diploid, that's not necessarily accurate. Most things that are like us are diploid. Most of the things that we call the land plants are diploid. But bacteria, archaea, fungi, protists, more haploid than diploid. So again, that's one of those misconceptions. Everything has a biology like us. No, not really. Are there any questions about fungi? Okay, if not, I'll leave this and I'm going to let you go for today. Tomorrow, we're going to start looking at different types of fungi and how those different types of fungi reproduce. And we'll get a firsthand lesson in what I mentioned earlier, that for every group of fungi, the spores have a different name, and the structure that makes the spores has a different name. And similar to what we did with Protista, what we've been doing really the last couple of days, we're going to see a lot of pictures. So if you download the notes for the fungi, don't get psyched out. A lot of these pictures are similar to what we would be seeing if we were looking at slides of these fungi in lab. If you do have a mushroom at home, you might want to save a mushroom for a day or two, because we will talk about mushrooms and you'll be able to see some of the structures that we talk about. So I'm going to stop sharing. Are there any questions before we leave? If not, I'll let you go today. So enjoy the rest of the day.
Excuse me, sir.
Go ahead, surprise.
Okay, yes, thank you. Did you receive my assignment? I submitted through the D2Hell mail.
I have to check. I don't know if I saw it through D2L. I think I might have just seen it in my regular email though.
Okay, I want you to help me evaluate it. I want you to help me evaluate it.
Okay.
Thank you.
You're welcome.
All right, bye, Mr. Griffith.
Bye-bye. See you tomorrow.